Hereby I open this academic ceremony in which Hala Ajjour will defend the academic thesis, Mitochondrial Function in Aldosterone-Dependent Human Hypertension. May I invite you to present a summary of your study and the conclusions of your thesis.
Highly esteemed Prorector, members of the Corona family and friends, thank you for being here today. It means a lot to me. In the next 15 minutes, I will be giving you an overview of what I did during my PhD thesis. If you look at the man in this picture, he looks healthy and he feels fine. However, he is still at risk of hypertension. Did you know that hypertension has no symptoms and no warnings? And usually it's known to hide until it strikes. That's how it earned its name, the silent killer. It is the leading contributor to cardiovascular diseases, #1 killer in the world and for kidney damage. And it's known to affect 1.3 billion people worldwide, being a global burden to patients and healthcare systems. In many cases, the exact cause of hypertension is not known, and in this situation, these patients are diagnosed with primary or essential hypertension and prescribed a lifelong medication. However, luckily, in other cases, the underlying cause is detected. That's when it's called secondary hypertension. And a common form of it is primary aldosteronism. That's when there's an excess aldosterone hormone secretion from the adrenal gland. Interestingly, 30 to 50% of these cases are what's called aldosterone-producing adenoma, which is a tumor or a mass only in one adrenal. The good news? This is surgically cured by an adrenalectomy. So how does aldosterone increases blood pressure then? It goes back to work on the principal cells of the kidneys. It causes salt retention, which is followed by water retention. This will increase blood volume, causing more pressure on the vessels, leading to increased blood pressure. In normal cases, aldosterone production is primarily regulated by the ROS system, and mainly by its last component, the angiotensin II. In this manner, angiotensin II binds to the AT1 receptor on the zona glomerulosa cell. This will increase intracellular calcium from the intracellular calcium stores. Not only that, it will also inhibit potassium channels. This will depolarize membrane potential, which will open voltage-gated calcium channels to lead to more intracellular calcium increase. Now, calcium plays a central role in aldosterone production in this manner. It facilitates cholesterol transport to the mitochondria, the main site of aldosterone production. It boosts NADPH production, and mainly it upregulates the key enzyme CYP11B2, all leading to aldosterone production as critical steps. So what happens in APA then? Research has shown that there are somatic mutations in ion channels or pumps that lead to either depolarization of the membrane potential of the zona glomerulosa cell or to direct increase of intracellular calcium, both leading to autonomous overproduction of aldosterone. So despite the well-established role of calcium in aldosterone production, the calcium dynamics in the zona glomerulosa, specifically in APA cells, have never been studied till now. So aldosterone effects goes beyond raising blood pressure or causing renal damage. It was also associated to cause arterial stiffness and calcification. Now, it's important to say that calcification process is completely normal and needed in hard tissues like teeth and bone, and it's pathological when it takes place in the vessels. What was shown clinically and precisely using CT scans is that primary aldosteronism patients have a higher abdominal aortic calcification when compared to essential hypertensive patients. Having all that said, the research aims of my project is to investigate intracellular calcium signals and mitochondrial reactive species production in aldosterone-producing adenoma cells and compare them to adjacent normal cells. We also aim to investigate vascular calcification propensity in vitro of APA patients' plasma. So to start, we recruited patients. These patients were diagnosed with APA precisely, with primary aldol precisely with APA, and they were referred to adrenalectomies. We received the specimens, and then we precisely selected CD56 positive aldosterone-producing cells from both normal and APA tissues. To follow instantaneous calcium fluctuations, we used live confocal microscopy. Now, to analyze the data, we designed a custom-made calcium detection pipeline that allowed data extraction from the live confocal microscopy raw images. And we had an output of calcium traces with detected single calcium spikes that they were also quantified in terms of 3 parameters. So by looking at calcium levels, and specifically at basal levels, APA cells had shown to have a reduced baseline calcium levels in comparison with normal cells. And both cells had shown to have spontaneous calcium transients that were also similar when compared. Now, to provoke calcium signaling, we stimulated cells with angiotensin II. The first calcium transient was higher in amplitude in normal cells, but as these cells continued to oscillate, they were higher in amplitude in APA cells. So after also observing that there was mainly 2 calcium phenotypes, an oscillating and ionic, we quantified the number of actively oscillating cells in both cell models, and we found that they were significantly higher in APA cells. Not only that, but also the frequency of oscillation was higher in APA, indicating that high aldosterone hypersecretion or production is probably regulated by calcium oscillatory activity. In the next part of my thesis, we investigated the effect of the TASC2 potassium channel on calcium signals. So TASC2 potassium channels were shown to be down-expressed in APA cells, and they were also associated with high aldosterone production. So what we did is that we inhibited these channels in normal cells, and then we quantified calcium signaling. At baseline level, calcium levels were not changed. However, inhibited cells, showed higher spontaneous calcium oscillations that also had higher amplitude. And when these cells were stimulated with angiotensin II, the oscillatory activity had higher amplitude. However, the oscillatory activity, or the number of oscillating cells and the frequency of oscillations, were intact, indicating that TASC2 channel inhibition doesn't fully mimic APA profiles. Now, to the next part of my thesis, we looked at the cytoarchitecture of the zona glomerulosa tissue that is responsible for aldosterone production. As you can see, these cells arranged in glomeruli or rosettes. And when we cultured our cells that were dispersed in vitro, after 24 hours, they were able to reaggregate, forming these multicellular clusters again. Not only that, but also after 96 hours, these clusters were getting bigger. So using statistical approaches, we showed that these cells contain subclusters of cells that have highly coordinated calcium activity. We also observed that cells within the same cluster showed a strong coordination than those in other clusters that are present in a different proximity. And importantly, tumor formation in APA did not disrupt the coordinated activity, as the pattern of coordination was similar in APA and in normal cells. To the next part of my thesis, we aim to investigate the reactive oxygen species production. And for this aim, we use the dye Mitosox, which precisely targets mitochondria and reacts specifically with the superoxide ions. When we quantified the results, we showed that APA cells showed a reduced initial or baseline reactive oxygen species when compared to the normal cells. And even after stimulation with angiotensin II, APA cells continued to show a lower reactive oxygen species production in comparison to APA cells, to normal cells. Now moving to the last aim of my thesis, in which we aimed to assess calcification propensity of plasma from APA patients using the cell-based assay, the biohybrid system, in which it integrates the circulating biomarkers in vascular calcification propensity assessments. We exposed the induced vascular smooth muscle cells to plasma from APA patients. By day 6, we were able to detect deposition of calcium phosphate crystals that are shown in red here. And after quantification of these results were clear. Plasma from PA patients showed a higher calcification propensity when compared to control cells. And importantly, plasma from essential hypertensive patients indicated less calcification propensity. These data indicates that PA plasma promotes more vascular calcification when compared to essential hypertensive and controls. In summary of my thesis, we were able to conclude that APA cells are characterized by increased intracellular calcium oscillatory activity and frequency of oscillation, leading to aldosterone production increase. While task 2, potassium channel inhibition, increases calcium oscillation amplitude, it doesn't fully recapitulate the intracellular calcium profile shown in APA cells. Aldosterone-producing cells naturally form clusters that exhibit coordinated calcium activity. APA cells may reduce mitochondrial reactive species as an adaptive mechanism to support sustained aldosterone production, and eventually induced vascular smooth muscle cells offer a non-invasive, personalized tool to monitor vascular calcification in PA patients. And with that, I would like to give the word back to the prorector.
Thank you for your presentation. The opposition will be opened by Professor Kroon, Professor of Vascular Internal Medicine at this university. Professor Kroon was also the chair of the assessment committee.
Dear candidates, to be honest, I was impressed by your studies, and especially the novelty of your PhD thesis. I read it with pleasure, and it gave me several new insights on hyperaldosteronism. Especially when, and you illustrated this very nicely again, the complex cascade that is going on when angiotensin interacts with the angiotensin II receptor, etc., and the aldosterone production. I want to congratulate you, but also your supervisor team, both in the Netherlands and Italy. Having said that, I want to go into the content of your thesis, and I'm not an expert in calcium signaling, but, well, I'm a clinical expert on hypertension, and you gave me, with proposition 5, immediately my first question. But before that, before my first question, I want one of your paronyms to read proposition 5 aloud for me.
Proposition 5, the use of calcium channel blockers to investigate intercellular calcium oscillations offer valuable insights into PA pathology.
Thank you. So calcium antagonists block the entry of calcium into the cell, and you nicely showed that it's necessary to start up the aldosterone production. So what would be then that valuable insight in PA treatment with calcium antagonists? And my question is, because we know that on several situations, especially when we do a salt loading test, we stop calcium antagonists. We stop amlodipine, nifedipine, because they increase aldosterone. Interesting, and well, you hint that this might decrease aldosterone production. Could I ask your opinion about this?
Sure. So to start with, let's go back to the aims of this thesis. If we looked at what increases aldosterone, we can see that in PA, RAS system is down-regulated because of the increased aldosterone production. And what actually increases aldosterone production in these cases, and specifically APA, are the mutations. And these mutations were shown to increase calcium. But it wasn't shown how they increase calcium in terms of calcium dynamics. And that's why we wanted to investigate calcium dynamics in terms of baseline calcium levels, in terms of calcium amplitude, duration of the calcium wave, the total area, but also the frequency of oscillations. And in terms of frequency and number of oscillating cells, they were constitutively and maintained in a maintained way increased in APA cells. And this was the first novelty of the thesis. So next we come back to this proposition. We didn't actually inhibit calcium voltage-gated calcium channels to see the effect on calcium signaling. And that's why this point is important. Because what actually open up these channels when there's no direct mutation taking place in one of them is the increase or the depolarization of the membrane potential. And usually we also have to mention that we have two types of these voltage-gated calcium channels, the T type, which are the low, and then the L type, which are the high. So this proposition takes a note or highlights the importance for further investigation on inhibiting these calcium channels to see if this will decrease the calcium activity, oscillatory activity, or the frequency of oscillation.
But in fact, what you're saying is that the mutation is more important than the calcium influx.
No, we're saying that they are definitely related, but what's important is how these temporal waves of calcium change. Because if we show that the frequency themselves and the amplitude of oscillatory activity are probably the leading cause for increased aldosterone production, if we try to inhibit these calcium channels, this will lead to less increase in intracellular calcium, which might decrease aldosterone production. Now, if we want...
But I'm still puzzled because several calcium antagonists, amlodipine, nifedipine, I told you, increase aldosterone production in clinical situation. Whereas others, verapamil is another calcium antagonist, decreases aldosterone production. So I'm puzzled with that calcium antagonist and the calcium influx, and the prevention of the calcium influx. And then if you say that the mutation is more important, or it has an important value. I missed what I'm. If that mutation is important, what? Well, I missed my next question. Well, never mind. Let's move on because I have an additional question on medication, if I'm allowed to, of course, because you're not a clinician, but still, I'm puzzled with the effect in the end in the clinic, and that's ARBs.
Yeah.
And you nicely said that ARBs, you showed that irbesartan decreased the oscillation or stopped the oscillation, the calcium oscillation.
Yeah.
Does that mean that the ARB is a treatment for primary hyperaldosteronism?
Okay, thank you for your question. Now, irbesartan in my thesis was used to indicate that angiotensin receptor is the one that is responsible for the increase. AT1 receptor is responsible for the increase of intracellular calcium. And since when we inhibited this receptor, we completely abolished the first transients and the oscillations. So the use of the irbesartan was solely to show the molecular pathway which receptor is implicated in these signals, which is AT1R. To go to your next question, in PA patients, the RAS system is downregulated because these mutations lead to increase of aldosterone production. When this aldosterone production is highly present in the circulating plasma, this will decrease renin. It will also decrease eventually angiotensin II production. So I don't think ARBs are correcting PA because PA, when there is a mutation, it's autonomous of the RAS system.
Okay. Well, there are still some puzzling effects I want to discuss with you, but time doesn't allow that, so I want to give the word back to the prorector.
Thank you. The opposition will be continued by Professor Dallas, Professor of Cardiovascular and Metabolic Health at the University of Glasgow, Scotland. Professor Dallas was member of the assessment committee, and we highly appreciate your participation on site to this academic ceremony.
Thank you very much, Prorector. Candidate, thanks very much for the very nice presentation. I must say, I have never seen somebody presenting the complexity of primary aldosteronism in slightly less than 15 minutes, so very well done. I also really enjoyed reading your thesis, especially the printed version, which looks really lovely. However, there are a few questions that I would like to ask you that are then related to your proposition number 4, but let's start with some general questions first, and they are clinical in nature. You give us a lot of detail about the results that you get in individual patients, but you do not present much of their clinical characteristics. Can I ask you, what kind of patients were these? Were they male, female, old, young? Were they on medication? Can you give me some insight, please?
Highly esteemed opponent, thank you for your question. Actually, we definitely have to mention that it's always highly important to show the clinical data or the biochemical cure of the patients when we're doing translational research. And that's the soul of our research anyway. So to give you an indication, these patients had a BMI of 26. They were 53% males, and they were all Caucasian, except for one Chinese man. They had an average age range of 57 years old. They started from 37 to 73. And I would also like to give you an indication of the mutational status of these patients. So, as expected, more than 50% of these patients had a KCNJ5 mutation. Now, why it's expected? Because KCNJ5 mutation, which is a mutation targeting the inwardly rectifying potassium channels KIR 3.4, are predominantly present in APA. So, we expected to see more than 50% of KCNJ5, but we also saw CACNA1D, CACNA1H, so 14%, 7%. There's also an ATP1A1, 7%, and CT and NB1 mutation, 7%.
Okay, so that's really helpful to understand this. And you probably know why I'm asking this question. You discuss it, and we see it in the data, obviously, that there was quite a variability in the results. you have, starting from baseline calcium signals to the calcium spikes that you see in amplitude and frequency, you see variability. Is this variability related to the variability that you see across the clinical characteristics of your patients?
Thank you for your question. So we definitely have to indicate the variability we saw. And specifically, these variabilities were in the parameters of these oscillations. So the amplitude, the area under the curve, and the duration of these calcium signals. And this is expected because we are grouping all patients coming from different age range, from different background of mutation, leading to these APA, but also what's also usually expected in hypertensive patients. These patients were already on different medications, and we don't know for how long it took for clinicians to diagnose PA. We don't know the drug history of these patients, so that all might affect these results. But these variabilities we did see in these calcium parameters were not seen in the frequency, and they were not seen in the oscillation activity. And this gives strength to this piece of data, to the frequency, because these were sustained. So despite all these factors that might induce variability in the results, frequency and percentage of cells showing oscillatory activity remain maintained lower or higher in APA, but also regarding the baseline calcium activity, we always saw a reduced in APA cells.
That's a really helpful clarification. Thanks very much. Can I just ask one technical detail that is about your calcium measurements? You present arbitrary units a lot, which is perfectly acceptable, of course. Can you tell me about the quantification here? If there's an arbitrary unit of 1.5, does this mean that it's twice as much calcium as with an arbitrary unit of 0.75? Or is there a non-linear relationship between the absolute value and the arbitrary units?
Thank you for your question. So we added arbitrary units just for the clarification, but this takes us back to how we did this calcium assessment or quantification. We use Fura2. Fura2 is a free calcium detector, and it's well known to be potent because it's a ratiometric dye. It takes into consideration any fluctuations or differences in signals that could be taken due to different specimens or different days of experiments, or probably different loading of dyes that we might accidentally do. So this dye has two emitting signals, 340 and 380. And as we also saw in these images, we have a red image, confocal image, and a green one. What we used first is to normalize each signal from each to the background, and then we had a ratio of 340 to 380. So I would say, so these are the results. The arbitrary units are equal to 340 to 380 ratio. So it would be, it's more like a linear effect of this increase.
Okay, I believe you this. And then in the interest of time, may I just pick one or two words from your proposition number 4? And that is actually where you propose that you have a new system of a vascular cell system that allows us for better precision medicine. Just in half a minute, what can I as a clinician learn from your data now? Can I translate them really into practice, and how would this look like? You can read the proposition if you like, but...
Yes.
So, proposition 4. Induced vascular smooth muscle cells are a novel and indispensable stem cell-derived model for personalized medicine to diagnose and treat vascular calcification.
And you can keep your answer very concise, please.
Yeah. So, vascular calcification leads to cardiovascular complication. It's an additional factor of cardiovascular complications. In case of PA, we already have a hypertension that is causing, that is targeting many organs, but also aldosterone itself. So if we were able to easily, when PA is diagnosed, to also assess vascular calcification, that would relieve the patient, because we are giving him an extra factor of relieving the aldosterone mediated effects on the body, which is vascular calcification.
I'll keep this in mind. Thanks very much. So I'll hand back to the prorector. Thanks very much.
Thank you.
Thank you. The opposition will be continued by Professor Fernandez Alfonso, Professor of Pharmacology at the University of Maastricht. Professor Fernandez Alfonso was also member of the assessment committee, and also for you, thank you for joining this academic ceremony.
Thank you very much, Prorector. Dear candidate, it's a pity that I cannot be on site there, but still present in this defense. I have really enjoyed reading your thesis, which is really very, very clearly written. Also the presentation, which I also agree has been really clear for in this short time, 15 minutes. And I would like to stress that you have used a wide range of experimental methodologies, which really implies an excellent PhD training, which is really mandatory for a doctoral student. So I would like to stress this, and also to you and to your PhD supervisors. So I have several questions, and these questions are more related to basic research. And I would like to start with the superoxide anion, or the oxidative stress in the mitochondria. So mitochondrial oxidative stress is a really complex process. You have assessed the ROS production, mainly superoxide anion production by mitosocs. How would you proceed to deeper assess what is going on oxidative stress alterations in APA cells?
Highly esteemed opponent, thank you for your question. Now, if we go back to the reactive oxygen species studied, we could see that these results were opposed to what we expected. Angiotensin II, when they were stimulating cells, ROS or superoxide was downregulated in APA. Now, this takes us back to what we know on this topic. We do know that angiotensin II usually increases reactive oxygen species. And we also know that angiotensin II doesn't have a huge role in aldosterone production in APA, because that's driven mainly by these mutations. So this and additional proposition that possibly APA cells has an adaptive mechanism to reduce ROS could explain what we have. Now, what we can do next is to definitely increase PA patients number. In these experiments, we only used 3 patients, 3 paired tissues, normal and APA. And we definitely need to recruit more patients to do so. And possibly we can check also other ions to check the NADPH oxidase activity that's most probably leading to superoxide ions, and possibly explaining the results we got. So mainly our aim is to increase the patients recruited for these experiments.
Mm-hmm. Thank you. And could you speculate which compensatory mechanisms might be acting to reduce this superoxide anion, both not only stimulated with angiotensin II, also in the basal APA cells?
So it's most probably related to the NADPH oxidative oxidation, and that's because NADPH is needed and essential for aldosterone production. So that's why probably that's taking from the superoxide that is being produced, because NADPH is being used for aldosterone production. But that's our main conclusion. It could be that to maintain the tumor formation in APA, cells do produce less reactive oxygen species. We need to check more what are the processes possibly protecting these cells.
Mm-hmm. So you haven't thought about the superoxide dismutase or other enzymes related to the antioxidant defense being altered in the situation?
Yes, and also we had an idea that could be technical. We thought that possibly the membrane potential of APA cells is dysregulated, and that's why possibly there's a down uptake of this dye from the mitochondria, because mitosox targets mitochondria depending on its membrane potential. So we also applied a TMRM assay, which is an assay that could give us an overview or insights of the membrane potential of the mitochondria in normal and APA cells, but the results were not conclusive, as it's written in my thesis. But that's also possibly because we only had 3 patients. So more patients are being recruited to strengthen these 2 experiments done already.
Yeah, thank you. And then another question is related to the AT1 receptors. You have nicely shown that irbesartan blocks calcium currents. My question is, what is your thought on the possible role of AT2 receptors?
Yeah, so AT2 has a protective role as what's known in opposition to AT1. So to give a background on this, AT2 is known to have this protective role, even though in some research papers also done by our group, it didn't show when that one was downregulated, it didn't show to decrease aldosterone production. So this is still controversial, but it would be a very interesting point to investigate for sure.
Mm-hmm. Well, thank you very much. Congratulations to you and your supervisors. Thank you.
Thank you. The opposition will be continued by Dr. Cecchetto, Associate Professor of Biochemistry at the University of Padua, Italy. And Dr. Cecchetto was also member of the assessment committee. And also thank you for you to joining this ceremony.
Thank you very much, Prorector. Dear candidate, first of all, congratulations on your thesis and the important work that you've done. Your project covers many areas from cell biology to mitochondrial function and even vascular complication like calcification. So, and also your presentation are well structured and clear. I'm a biologist and since my expertise on ion channels, I have some questions about TAS2 inhibition, a question about calcium mitochondrial interaction, and a question about genetics and functional interpretation. So starting from the first question that is about TAS2 inhibition, I have a question about the blocker of this channel, because the blocker that you use is not specific only for TAS2 at the micromolar doses. So I want to ask you if did you consider more selective inhibitors of this channel like A1899 or genetic knockdown approach to validate the role of TAS2?
Opponent, thank you for your question. No, not really. We did not use any other drug, but it would definitely be interesting to also test other drugs and see if they have the same efficiency. In terms of genetically modified cells, we cannot do that because our main focus in this research paper is to use primarily cells. So there's cells that are freshly taken from patients and that can reflect what's actually happening in the physiological or pathological levels in patients. But it would also be interesting to mimic these, possibly in HAC15 cells. And test if we could also get the same results of APA phenotypes. I would also like to mention here that if this is the purpose, it's very important to know the cell model we're using, because we need to use cell lines coming from patients. Otherwise, if we're looking into using mice cells, for example, mouse do not express TASC2 at all. So it's always important to also check the cell model we're using.
Okay, thanks. But related to these things that you use the APA patients-derived cells. It is also true that in these cells, there are a lot of somatic mutations, not only of TASC2, but also KCNJ5 or ATP1A1 and so and so. These somatic mutations are known to affect the ion homeostasis and the membrane potential. So did you analyze the calcium imaging result based on genotype? So do you think, in other words, do you think that different mutation could lead to distinct intracellular calcium signaling? And these can explain some of the variability that you observed in the aldosterone secretion profiles?
Great question. So we definitely need to emphasize on the point that TASC2 doesn't hold the mutations. TASC2 is down expressed. And these mutations, if they occur in potassium channels, they target KCNJ5, so another type of potassium channel. That's for one. Another point is that mutations, no matter what the kind of mutation is, it leads to depolarization of cell membrane. And this is how we categorized our cells. They definitely have depolarized cell membrane compared to normal cells. And that's how we expected these cells to be comparable when we inhibit TASC2 channel. Okay.
The last question is related to the mitochondrial calcium uptake and the mitochondrial membrane potential and then the increased ROS species, because I have the curiosity if you consider the mitochondrial calcium uptake inhibitors or the idea to silence, for example, the mitochondrial calcium uniporter to test if calcium oscillation are directly responsible for these mitochondrial changes.
Thank you for your question. This part is actually very crucial. And if there's anything that should be done afterwards is this, because aldosterone takes place in the mitochondria. And calcium is shown to increase aldosterone production indirectly, but also directly by going in the mitochondria and exerting some effects like boosting an ADPH production. So why didn't we use that? Because that's challenging when you're using primary cells. You need a lot of number of cells to use, and at that time we were going strategically, starting from cytoplasmic calcium to go down to mitochondrial calcium, specifically after we saw that the basal calcium level is downregulated in APA, and that's how we hypothesized that probably it's a more buffering of calcium into the mitochondria in APA, which is leading to this piece of data. So definitely in the next stages, it's important to find ways to measure intracellular calcium in the mitochondria on primary cells.
Okay. For me, it's all. So thank you for your discussion and congratulations again to you and your supervisors.
Thank you. The opposition will be continued by Dr. Racing, Associate Professor of Biomedical Engineering at this university, and Dr. Racing was also member of the assessment committee.
Thank you, Mr. Pro-Rector. Dear candidate, first I want to congratulate you with your thesis. I agree with the others that it's really stimulating material, and I've looked at it from the system dynamics point of view. I would like to start with page 60, where you have a figure that's clearly demonstrating or showing us the oscillations that you find and the question around baseline. What is that? So let's start there. So my first question is, why the baseline shift? Is that an artifact of the measurement system? It might be in the image quantification or maybe the stimulus itself. Or is it a physiologically meaningful characteristic?
Highly esteemed opponent, thank you for your question. Actually, this was a main challenge in my thesis. It's to how to analyze these data, especially as you mentioned, the baseline after angiotensin II was most probably shifting either upwards or downwards, which gave us more extra challenge to know how to analyze this baseline. So if we go back to literature, when these experiments were done on bovine cells, baseline was also shown to not go back to the initial state. That could be possibly explained by the membrane potential of these cells, how these cells is regulating increase in calcium. This is a valid question, but it still has question marks in the studies. Even when studies were done on mice cells, we're still not sure how exactly membrane potential or potassium channels, in addition to voltage-gated calcium channels, are actually tight controlling the intracellular calcium increase in these cells. So it could be explained more by studying these conductances.
Okay. And also wondering about the oscillations itself. Maybe there's a bit of an answer there, but I don't know. I thought, as an engineer, how you built an oscillator. So you create a stimulus and you get a response, and you make sure that the response is fed back to the input, to the trigger, in some way, so that it gets into an oscillation loop. Now, in your presentation, you've also demonstrated where that loop might be. So can you describe the loop? And the oscillations are triggered by angiotensin II and to the receptor. So that's the trigger. So where is the loop that actually gets the system into oscillating?
Thank you for your question. So we definitely have to start that calcium fluctuations in the cell takes place as waves or oscillations, because otherwise, if it goes as bulk, it will be toxic to the cell, and cell will go into apoptosis. What could be explaining these are back to the conductances. So if we look back at the membrane potential of the zona glomerulosa, it's hyperpolarized at -80 millivolts, and then it goes up to -60, which allows the T-type calcium channels to open up. But at -30, that's where the L-type high voltage calcium channels are actually opening up, and then it goes down by potassium channels that work. In case of an APA, what's actually keeping these cells to oscillate, so what gives this loop, could be the mutations themselves. These mutations, with their continuous activity, could be dysregulating the membrane potential, and the initial state could be depolarized, high depolarized, more than what initially should be or what's normally should be, and that when the signal ends, it will restart again due to these dysregulated mutations that are dysregulation the initial membrane potential of these cells.
So you're relating the mutations to an overreaction or an oversensitivity of one of the channels in the cycle, which steers then the cycle to return back again into an oscillation. Is that what you mean?
Yes, it's more of dysregulation, the membrane potential, and what the membrane potential has as an effect on the conductances, on the opening and the closing of calcium, voltage-gated calcium channels, but also on the potassium channels that initially regulate these waves. Because that's what's different also between APA and normal cells.
Yeah. So then I would like to take that forward to looking at the rosettes and their behavior. You're describing the intercellular communication, also relating to the oscillations. So seemingly, it seems that the calcium oscillations, they kind of are a way of communication between cells. And I was thinking, might there be an explanation in that or actually the first question is, have you measured the activity, oscillatory response of a whole rosette? So in the image, you can actually take a larger region of interest and see what it does. And if you've done it, did it show no baseline issues? So basically, a clear return to baseline.
Thank you for your question. That's actually an interesting point to look at the data. What we did is that we took the individual cells' oscillations or signals, and then we tried to see if there's any coordination. And that's how we ended up seeing that there is a coordination, but in a subcluster manner. But we didn't look at a cluster as a whole entity and then getting one signal out of it. And if I want to correlate between your question and what we have, what we could possibly do is go back to the subclusters, have one signal, one representative signal of each subcluster, and then analyze this. It's a very interesting point to look into.
Yeah. So, I think that's a great idea.
Yeah.
I'm also thinking that if cells all have the same attitude, like if you describe a group of 3 people and they all look at the same, will they interact? No, they'll all have the same attitude. So if they have all a different attitude, so a different baseline or a different oscillation, you get interactions with it. That's more a conceptual part of that. So that's the reason why I want to look at it. All right. Last question. I think I have a quick question. So you described also, or basically your thesis says angiotensin 2 through the AT1 receptor has an effect on aldosterone, so basically on the ROS system, as well as on vessels, stiffness. Can you relate how that then gets you into subtypes of hypertension? So how do you relate these 2 pathways or these 2 mechanisms related to Ang2 to systolic or diastolic hypertension?
Thank you for your question. So in PA patients, PA pathology is leading to increased blood pressure. And the increased blood pressure is not the only pathological concern in these patients, because as we saw.
If you say increased blood pressure, what do you mean? Dystolic or systolic or diastolic?
Yes.
Or both?
Both. So when patients are being treated, usually they need to be treated for PA. So the PA needs to be corrected, but also they need to have a correction for an increase of blood pressure. And if we want to relate these two topics, vascular calcification and oscillations, we would be focusing more on correction of PA and specifically on the increase of aldosterone and all the pathological molecular signaling that could be caused by aldosterone.
Okay. In the interest of time, I'm giving back the word to the prorector.
Thank you. The opposition will be continued by Professor Koenen, Professor Biochemistry of Vascular Inflammation and Thrombosis at this university. Professor Koenen is also the secretary of this degree committee.
Thank you, Prorector. Dear candidate, I would like to congratulate you with your thesis and of course also your team. I've read it with a lot of interest. I must say I am not an expert in the field, so but I still have some questions that raised and I would like to discuss with you. But before we start, I would like to ask one of the paronyms to read proposition number 2.
Proposition number 2, intracellular calcium signaling in human adrenal cortical cells exhibit a distinct profile compared to that observed in animal models, highlighting limitations in the translational accuracy of animal-based studies.
Yes, it's a very interesting proposition, and it is a little bit bold in my opinion. So before I would like to discuss this thesis, I would just like you to explain a little bit more how you came to this thesis and what the difference between your system and the mice might be.
Highly esteemed opponent, thank you for your question. I would like to raise up 3 points. First of all, we did see differences when we compared mice models to human models. In terms of potassium channels, human cells do express TASC2, potassium channel. And not only that, TASC2 was shown to be downregulated in APA, so it definitely plays a role in aldosterone production. That's for 1. 2, if we do compare phenotypes or calcium phenotypes between mice and the humans, we do see a clear distinction in these patterns. Mice shows lower spikes that they are higher in frequency, and I'm not sure if we compare amplitude how that's going to be. However, APA cells are much more heterogeneous, and if calcium, and they show different pattern. If calcium is a central key regulator of aldosterone, these temporal changes will be decoded differently by the calcium sensitive proteins like calmodulins, which will exert different actions. 3, if we go back to the research done on mice, when these normal cells, normal zona glomerulosa cells, were stimulated with angiotensin II with different doses, because that's how they compare the cells, they didn't have an APA, cells didn't show a higher frequency. Cells showed a higher burst of cells. So there was a burst of cell that there's a small time of no signals, with the same frequency, but different bursts. And that's how the aldosterone production got higher and higher as angiotensin concentration was higher. So we do see a distinction between these signals seen in mice and these signals seen in humans, and that raises many questions.
Okay, so it's about the repertoire of receptors and their behavior. Mm-hmm. So just to be clear, in mice, you mean in mouse cells, or have you looked in mice at the site using, for example, an intravital microscope?
In murine cells based on literature.
Okay, because what I was thinking of is, of course, the physiologic environment of the cells, the way you culture them. And I've heard, and you've already talked about rosettes in a previous question by the opponent. So I would like to elaborate a little bit on these rosettes. So the rosettes are always present in your cells, in your cell culture? So all the cells are rosettes?
So before answering this question, I would like to say something about rosettes. Rosettes are completely normal in cells in the embryogenesis, and they are only sustained in the zona glomerulosa tissue postnatally. Mm-hmm. That's for 1. When we looked at these cells in vitro, we did still see some isolated cells that were alone.
Okay.
So not all cells were able to reaggregate and form these multicellular rosettes or clusters.
And the clusters that you did see, did they also show differentiation towards, I think it's zona fasciculata or something?
Yeah?
Yeah,
Okay.
did they also differentiate?
Yeah. Okay, so to tell everybody, these rosettes or these glomeruli in the intact tissue of the zona glomerulosa are dynamic, and as they form, they also resolve. And naturally, they need to resolve into zona fasciculata and zona reticularis, and that's how the zona glomerulosa in the intact tissue renew itself. So that's how this question is coming from.
Yes. So before because that's very clear, but I want to give you the occasion, or want to give myself the occasion to ask the real question. So if you look at this, and you say it implicitly, heterogeneity of your cell culture. What does cell cycle state, differentiation, pairing of the cells, single cell, were they all comparable between the culture conditions you used when you did all the calcium experiments you showed in this thesis?
That's an interesting question, especially that in mice cells, after 24 hours or so, cells start to transdifferentiate. We didn't see that in these cells. What we do know is that the aldosterone production over probably 72 hours in these primary cells goes down. So that could also explain transdifferentiation. But we didn't look into that, and we didn't see it in a morphological way.
Thank you for your answers.
Hala Ajjour, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations and our return in this room.
All right.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. I know what's on the line. I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Make the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. South side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. Long road to the south side. 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
Hala Ajjour, the degree committee here present has assessed the quality of your thesis and your defense in view of its positive verdict and taken into account your previous qualifications. The degree committee has decided to grant you the degree of doctor. Professor Skurgers is authorized to confirm upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
All right, please.
Before saying the magic words, I have to ask you a question. Do you promise to work in accordance with the principles of scientific integrity at all times? To be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Hala Ajjour, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate, signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of our university. Glasses again. Dear Dr. Ajjour, dear Hala, I'm the first that is able to congratulate you from this place on this fantastic achievement. A great presentation we have seen today, a great booklet, and also a perfect defense. You achieved now the highest academic degree that is possible. In these congratulations, I also want to congratulate your supervisors from Italy, Professor Rossi, who unfortunately is not present here today, but also Professor Lenzini. Thank you for taking the effort, coming here all the way, even needed to take a Flixbus yesterday to come from Amsterdam to Maastricht because our railway was on strike. I also want to extend my congratulations to your husband, to your family, your friends, and of course also your colleagues. I also want to thank first the members of the Thesis Assessment Committee, present here in person or online. From Maastricht, Professor Krohn, thank you for taking the role as chair. Dr. Reesink as coordinator of this fantastic consortium, thank you. Professor Koenen, thank you for joining and for stepping into a new arena as a research topic. A special thanks to the members from abroad, Professor Cecchetto from Italy, Padua, Professor Fernandez Alonso, thank you very much for participating. And last but not least, Professor Dallas, coming from Glasgow, and also that was a challenge. We even had to have a phone call yesterday. You were actually close to boarding and then saw that there were no trains going to the south of Maastricht. So you were very flexible, took a hotel, not in Maastricht, but in Amsterdam, and this morning, 6 o'clock, you were the first in the train coming to the south. Thank you very much for taking this effort and being here present. Dear Hala, you are one of 15 ESR's early stage researchers in the European Horizon 2020 Mindshift. This collaboration between Maastricht, Glasgow, Paris, Madrid, and Padua started officially the 1st of January 2021. Within Mindshift, the aim was to train a new generation of young researchers in understanding the dynamics, interactions between vascular and endocrine pathways in hypertension. Mindshift is a network of colleagues and friends, which form the basis of this great success. The 15 ESRs were selected from all over the world: Belgium, China, Bulgaria, Ghana, Vietnam, Iran, Italy, Pakistan, Rwanda, the UK, Mexico, and finally 3 ESRs, including you, from Lebanon. So I think that the education in Lebanon is really, really great. A wonderful group of young, talented researchers trained in a double joint doctorate training program. And within Mindshift, your project significantly contributed to this success. The supervisors for the ESR number 12 were Professor Rossi, Dr. Lanzini, and myself. And it was no surprise that you were chosen as the candidate for this position. Yes, you were definitely the best candidate that we interviewed. But interestingly, you initially missed the interview because you had missed the email with the date and time to have the interview. After calling you, where you were, we could do the interview. And apart from this initial sign of absence, you gave a brilliant interview which made us confident in choosing you for the ESR position. And you officially started in October 2021, which indicates that you finalized your thesis within 3.5 years from your starting date. A really fantastic achievement. Hala, you are very practical and hands-on in the lab. You integrated well in the team and contributed actively to the experimental work. Over the 3 years of your PhD, you developed a strong sense of independence and became capable of managing your own work. You worked at a steady pace in the lab in Padova. But then you had to come to Maastricht, where you were forced to work with different cells than APA cells, these vascular smooth muscle cells. And you were challenged to culture both primary smooth muscle cells and also the iPSC differentiated smooth muscle cells. And these cells are a little bit like little children. They ask attention all day, even early and late hours, and even weekend days. And since you worked very precise and meticulous, you had to compensate that with hard work and very long hours, also forcing Amor staying very long in the lab. And indeed, we have to get you to know you as a very hard worker, who listens to advice and also, importantly, follows this advice. Also with the finalization of your thesis, your propositions, and the presentations today, we had a lot of back and forth on that, which made that you are a very dedicated, proactive, and fast in response person. There are also some fun facts about you. You married during your PhD period. And what a story is this? Normally, these ITN consortia provide a PhD position, and at the end, like today, you earn your title. But for you, it was more. When you started your PhD position in Padova, arriving from Beirut in Italy, you were picked up from the airport by an unknown person for you back then. And he was Lebanese. And he was so nice that he is now your husband. What a fantastic story is this. Lebanon, a country we talked about a lot. I know you miss it very much. Your family, your culture, your country, as well as the food. I had the luxury to visit Lebanon several times during my scientific life. And I could experience and witness this great and beautiful country with the best kitchen in the world and with very great hospitality. I hope, Hala, that we will one day meet in the future in Lebanon. Hala, finalizing. It was a great pleasure working with you, and I wish you all the best for the future. I'm pretty sure you will succeed in whatever you are going to do. For now, the thesis defense is over, the stress is gone, you can relax. Enjoy and celebrate this fantastic achievement and moment.
I would like to...
No. Dear Dr. Ajjour, also on behalf of the Board of Deans of the Maastricht University, it's a pleasure for me to congratulate you with this new degree of doctor. The University of Maastricht, and I assume also the University of Padua, wishes you all the best in your future scientific career. So hereby I will close this academic ceremony. I will ask the audience maybe to move already to the reception room to congratulate the new doctor, and we will make a picture here in the aula in the presence of the online participants and opponents and the new doctor, and thereafter we will make a picture on the stairs in the central hall. Hereby I close this academic ceremony.